Thank you for the introduction and sticking around for the last presentation. Today I will present on the thermal reactivity of proteins in peanut seed during roasting as identified by proteomic analysis. Peanut flavor is a complex matrix and it's impacted by a variety of environmental and processing variables. It's been reported that the seed storage proteins are about 25% of the proteome and impact important aspects of the seed's performance, including flavor. Peptides and amino acids provided by the seed storage proteins react with carbohydrates to form flavor compounds. And these flavor compounds include pyrazines, furans, and pyrroles, to name a few. To better understand the roasting process and generation of flavors, this project sought to identify the specific amino acids and peptides that react with sugar and quantify them. Also, we thought we could find the specific protein precursors that serve as the raw materials, sequence them, and identify them. Proteomics was selected as a potential analytical tool to do that. This made us ask the question, can a bioinformatic approach be utilized to generate a global picture of flavor-producing reactions during roasting? In order to do this, we needed a method to prepare samples for proteomic analysis, a collaborating lab to run the samples, methods to analyze the data and compare data sets. Developing the method would allow us to compare roasting processes and variables in flavor production. Each part of the omics research picture, genomics, transcriptomics, and proteomics, can provide a different picture and a partial picture. Proteomics provides an overview of protein components expressed in the seed, as well as any post-translational modification and reactions that occur during physiological responses to environment and other factors. In order to accomplish these goals, we prepared samples for traditional sensory analysis and gel electrophoresis alongside proteomic analysis at different roasting time points and color targets. Peanut roasting, blanching, and sensory analysis were conducted according to published methods. Samples were defatted according to our lab protocols. Defatted samples were separated by SDS-Page electrophoresis. We either ran the gel for a limited time and pooled all of the soluble protein, or we ran the gel to completion for better resolution of individual protein bands. The gel band or bands are excised from the gel and shipped overnight with temperature control. At the Proteomics Resource Center of Rockefeller University, samples were digested with trypsin, desalted, and analyzed using LC tandem mass spectrometry. The spectra were extracted using MaxQuant software and queried using the Perseus proteomics platform. That platform provides statistical tools for high-dimensional omics data analysis covering normalization, pattern recognition, and time series analysis. Once the data was generated, we treated, sorted, and classified the data, developing models to help visualize trends. First, to confirm the thermal breakdown of proteins in the defatted fraction, we performed gel electrophoresis on the roasting series. On the left-hand side, you see an image of this gel starting at the unroasted fraction, and then 8 minutes, 10 minutes, 12, 14, and 16 minutes. As visualized on the gel, as roasting time increases, the size of the bands decrease and resolution improves. We are also reporting the corresponding sensory data on this set of samples. On the right-hand side are the panelists' average scores for roasted peanutty, raw beanie green, dark roast, sweet, and bitter flavor attributes. As expected, as roasting time increases, the roasted peanutty trait steadily increases to a maximum value. Raw beanie green decreases, coffee dark roast increases. The sensory checkpoint ensures that we are capturing samples from the critical portions of the roasting process, including under roast, target, and dark roast. On the right-hand side is a picture of the representative samples sent for proteomic analysis, depicting the color development during the roasting process. At the top would be 8 minutes, and then increasing roasting as you go down. The results can be summarized as follows. The unroasted seeds contain nearly 1,400 identified proteins, which were fragmented into over 11,000 peptides. We queried the sequences to identify the proteins and cross-referenced 3 different databases listed there, GeneBank, UniProt, and PeanutBase. Multiple reaction types were identified and reactive amino acid sugar combinations noted. We identified the types of amino acids most reactive during the roasting process. We selected the 5 most reactive amino acids, added sugar, and roasted them in the test tube. The results presented demonstrated that these amino acid sugar combinations tested in the lab produced flavor compounds similar to roasted peanuts. That particular presentation was given at APRES in 2019 by Matt Skolton. Right, this chart shows the data for 17 peptides, giving the relative abundance across the different roasting time treatments. We identified the most reactive and abundant peptides. In this particular set, showing at the 8-minute mark, the abundance is shown all in green. And what we believe to be happening here is that this initial heating event helps the protein unfold and exposes more parts to the tryptic digest in this case. Then, as that protein was exposed, as time of roasting increased, more and more of the protein could react. So, in other words, first the proteins unfold, then amino acids on that peptide reacted, and the parent peptide was eventually completely broken down into flavor compounds. We took all of the peptides and plotted the data. This is an example of 2 of the peptides out of the set of 17. On the y-axis is the relative abundance, and on the x-axis is the roasting time, beginning at 8 minutes and continuing to 14 minutes. This information was used to characterize the relative reactivity of each peptide and confirm the correlation between amount and roasting time. At this point, we began a deeper dive, looking at the number and type of reactions for each amino acid in the peptide sequence. Then the protein sequences were reconstructed and all reactivity data layered on top of the sequence. For example, this graph illustrates the compilation of sequencing and reactivity data. On the y-axis is the number of reactions, and on the x-axis, the amino acid position. This data represents one of the most abundant proteins in the seed that is also highly reactive during roasting. The peptides were sorted and ordered according to sequence. Each amino acid reaction, and considering there are multiple reactions and reaction products, were counted and plotted versus the ordinal position of the amino acid in the protein sequence. All the different roasting treatments are shown in different colors, from raw all the way to darkly roasted. And if you look at the peak around amino acid number 400, you can visualize how the amino acids in that section of the protein react as time increases. So we can know which amino acids are producing flavor. This information provides a fingerprint of the roasting process and demonstrates how thermal treatment influences the reactivity of the raw materials. From here, we can compare different varieties, maturities, roasting technologies, etc. In conclusion, we report a method for analyzing the proteome in order to characterize the impact of roast on the raw materials of the peanut seed. We now have a working method for generating samples and analyzing data. This new tool can be applied to a variety of processes. And understanding how performance is tied to specific gene products and functional proteins allows us to apply the method to analyze differences in roasting processes, genotypes, and environmental variables. We would like to thank our collaborators at the Rockefeller Proteomics Center, and I've also included references here, and I will be happy to take your questions.
Thank you, Julie, for that presentation. Do you have one question in the Q&A toolbar button box here? At what temperature did you roast peanuts?
This was done at 350 Fahrenheit, which is approximately 177 Celsius.
Okay. Any other questions from our audience? Okay, Julie, thanks for being here today and for the presentation. We really appreciate it.